Nice outline. Could everybody please take their seats? Can people hear me okay? Is that okay? Okay. Okay. Can everybody hear me okay? Good morning. I'd like to welcome everybody to the conference today. Derek DeSilva can't be here right now because of a family emergency, so he should be arriving later today. We have Dr. Dorothy Petruka, who will be the moderator for the morning and afternoon, probably for most of the session until Derek is here. She is also a co-chair. What I'd like to do is recognize two distinguished guests that are in the audience. And I'd like to tell you these are in addition to all of the distinguished guests who have registered and the distinguished guests that are speaking and those that are exhibiting. We have Sandra Evans from the Armed Forces. Sandra, can you raise your hand? Back there in the white outfit. Yeah. Sandra Evans and the Armed Forces have been instrumental in a number of advances in Lyme disease, including the development of Permanone, as you know, the kind of thing that kills ticks on clothing. And Dwayne Gubler is here from the Centers for Disease Control. Dwayne, are you in the room right now? Can you stand so people can see you? We are glad to have you also. As you know, the Center for Disease Control has had a number of very large, well, I consider them very large grants. I suppose the scientific community would like a little bit more for both research and education. To let you know about the conference, although it looks a little sparse in here this morning, we have over 500 people in attendance that will be coming in and out. Some of them are physicians in the state, and they will be coming in and out during the days after they do their rounds. So please don't let any of the little activity that will be going on disturb you. Also, I'd like to tell you that no speaker is paid an honorarium for speaking here. There is no conflict of interest. That we have a reimbursement policy that reimburses for reasonable expenses, so we have no whining and dining and any special things that go on with the speakers. At most, we will probably give each person a video and a heartfelt thanks for coming and sharing their latest information and scientific discoveries. The speakers that come not only have world-renowned expertise and information to share, but they come because they want to network both with other healthcare professionals and the frontline patients that are out there. We admire them greatly because of the amount of effort. The first couple of meetings, all of our speakers paid their own way and registered, so there was quite an extraordinary dedication to moving science forward to the truth. I'd like to start out by introducing Dorothy Petruka. Dorothy Petruka is a board-certified pediatric neurologist with Cornell, New York, but most importantly, Jersey Shore Medical Center, who has given us our CME credit. Dorothy? Thank you very much. First, of course, welcome to Atlantic City and to this Lyme conference, which is 99.99% due to the hard work of Karen and Tom Forshner, who deserve a lot of credit for this. Jersey Shore Medical Center is in Neptune, New Jersey, which is in Monmouth County, about 70 miles north of here. And we're very proud to be part of this program and to sponsor it and to provide the CME credits. And on behalf of Dr. DeSpirito, our Director of Medical Education, I'd like to welcome all of you. Thank you. First speaker I'd like to introduce is Dr. Willy Bergdorfer. Wait a second. Dr. Willy Bergdorfer is a PhD, but also he has 3, maybe 4 now, honorary MDs. It's hard to keep track, and honors from around the world. The thing I'd like to tell everybody is that when we originally started the foundation, we called up Willy and we asked him to help us, to be on our board of directors. And Willy said to us something very interesting. He said, "It is an honor to be in public service, and the people we are loyal to are those that support us, the taxpayers. So I will help you at the foundation if you concentrate on getting information out to the public." And that was our first focus, and that has been our primary focus, is getting information out now first to the public and then to the scientific community. And we've expanded from there. But it's the true dedication of Willy Bergdorfer that has inspired us. I admire him greatly, as you may know. I think many, many people that know him do. He's our first speaker, so I'd like to introduce Dr. Willy Bergdorfer, the discoverer of Willy Bergdorferi. Have a good talk.
Thank you, Karen. Good morning, ladies and gentlemen. Dr. Petrucia. Karen, I have to tell you, the subject I'm going to discuss this morning is not exactly all about Lyme disease. And you have to thank Karen for that, because she said you have to talk about what these creepy crawlers are doing in this country, not only Lyme disease, and they do a lot of other things. So please bear with me. It's a slideshow. Relax, lean back, go to sleep, or whatever you want to do. May I have the first slide? Let's see whether I can put that on. I'm always afraid of that. I do. Is the juice on back there? Help! I said a long time ago we get people to the moon, but by golly we can get these projectors going. What am I wrong?
I think you had it on.
I had it on?
Yeah. There you go.
Thank you, Gilby. That's I like that. Thanks a lot. You have in your package a table that shows you the diseases that are transmitted by ticks in this country. And I very briefly refer, does this have to be on like that? Probably. To the tables: Lyme disease, Rocky Mountain spotted fever, tick-borne relapsing fever are the major ones. And then you got human babesiosis. You can in your room peacefully go through those tables and look at the agents, the tick vectors and their geographic distribution. Tularemia is still a problem. And then we got newly emerging diseases such as human ehrlichiosis, with Ehrlichia chaffiensis being the agent. We will hear that we don't know anything about the tick vectors in the southeastern United States. Colorado tick fever is still a problem throughout the Rocky Mountain areas, with Dermacentor andersoni being the only vector. Tick paralysis, you once in a while hear some cases of tick paralysis, not very many, maybe a dozen every year. Then we have canine ehrlichiosis, which is raising its head above the surface, with Ehrlichia canis, a disease in dogs. We got equine ehrlichiosis, a disease in horses. We got Potomac horse fever, Ehrlichia risticii. We don't know anything about the vector. Then we got anaplasmosis, and we got episodic bovine abortion in the western part of the United States. Very economically very damaging disease, with Borrelia coriacea as the agent and Ornithodorus coriaceus as the vector. Now all these listed diseases are zoonoses, whose causative agents are maintained and distributed in nature in a more or less complex developmental cycle between the tick vectors and their host animals. In many instances, humans, along with their domestic animals, become parts of these cycles either through accidental intrusion or as a result of prolonged sharing the biotopes with those of ticks and tick hosts. Thus, tick-borne relapsing fever, for instance, is acquired while spending nights in old log cabins. There's an example of it. Cabins that have become ecological parts of the spirochete Borrelia hermsii. hope you can see the spirochetes here in the blood. A spirochete that is normally maintained between its rodent hosts, chipmunks and pine squirrels, and the tick vector Ornithodorus hermsii, which is a soft-shelled tick as seen here. Similarly, the high prevalence of Lyme disease in certain northeastern and midwestern states is closely related to increased infestations by the tick vector Ixodes daminii, and Dr. Oliver, I'm going to tell the audience that you will rename that back to Ixodes scapularis. As a result of increased numbers of white-tailed deer, here is Ixodes scapularis, deer that serve as the host for the adult tick. Let me give you an example. In 1971, the deer population in Wisconsin, for instance, was estimated to be about 500,000. In 1981, 10 years later, the estimate was doubled. In 1987, the deer kill alone, according to the State Department of Natural Resources, was 250,000, and the estimated number of deer today is about 2 million. As the deer population increased and spread, so did the infestation and range of the deer tick. The example of tularemia should remind us that tick-borne pathogens are not always maintained and transmitted by ticks only, but possibly also by other blood-sucking arthropods such as fleas, lice, tabanids, mosquitoes, and so on. Similarly, there may be other means of transmission, including direct or indirect inoculation of the skin or the eyes during handling diseased animals, in the case of tularemia, the cottontail rabbits and water rats, also through ingestion of insufficiently cooked infected meat, or the consumption of water contaminated by urine or feces of infected animals. Such modes of transmission should be kept in mind, especially for newly emerging diseases for which a transmitting vector remains elusive. I'm thinking especially of Potomac horse fever or human ehrlichiosis, for which the modes of transmissions are still unclear. For many years, Rocky Mountain spotted fever has been the most prevalent and most important tick-borne disease in the United States. This has changed rather rapidly with the emergence of Lyme disease, a complex tick-borne spirochetosis that affects thousands of persons, children and adults alike. Lyme disease, as we shall hear later in detail, is a multi-system illness of the skin, the muscles, the joints, the cardiac and nervous systems, and to a lesser extent other parts of the body. Some of the clinical manifestations of this disease have been known in Europe since the beginning of this century under different names, such as erythema migrans, as you see here, tick-borne radiculitis, case of Bell's palsy, lymphocytoma benigna cutis, very rarely seen in this country, acrodermatitis chronic atropicans, also far more frequent in Europe than here. In the United States, the disease was first observed and recognized in 1969 in Wisconsin by Dr. Rudolph Scrimenti. Rudy, where are you? He is right here in the front. A few years later, erythema migrans was also described in a retrospective study of arthritis outbreaks among children and adults in the Connecticut community of Lyme, Old Lyme, and East Haddam. However, since arthritis is rarely mentioned in the European literature, the episodes in Connecticut were thought to represent a hitherto undescribed illness referred to as Lyme arthritis and later Lyme disease or Lyme borreliosis. The causative agent of Lyme disease, now known as Borrelia burgdorferi, has eluded investigators until the fall of 1981. You have heard the story about the discovery many a time, so let's just show the spirochete here in the midgut of the transmitting tick Ixodes scapularis. Because of variations in case definitions and because Lyme disease in many states is not a reportable disease, there has been no accurate data as to its prevalence. From 1975 through 1979, let's briefly recall, 512 cases were diagnosed in 14 predominantly northeastern and midwestern states. In 1982, the year the discovery of the spirochetal etiology was reported, 491 cases came to the attention of CDC. This total increased to 1,500 in 1987, and since then has been increasing steadily. Last year, in 1992, according to preliminary CDC counts, a total of 9,500 cases were recorded, which meant an increase of about 20% over the total in 1990. As in previous years, the majority of cases occur in the northeastern parts of the country. Of particular interest are the figures from Missouri, Georgia, and Texas, where in 1990, 205, 161, and 44 cases respectively were reported. In these states, the well-established vectors at that time, this has to be changed, Dr. Oliver, at that time it was Ixodes damini, is absent, and therefore it is speculated, or was speculated, that other species of ticks, such as the Lone Star tick, Amblyomma americanum, the female here, the male here, and or the dog tick Dermacentor variabilis, the bad picture of it, or perhaps other blood-sucking arthropods such as fleas or tabanids may serve as vectors. I am certain that Dr. Masters will bring us updates concerning Lyme disease in Missouri and the ticks that may be involved as vectors. I would only briefly emphasize that Borrelia burgdorferi or Borrelia burgdorferi-like spirochetes have been demonstrated in the Lone Star tick Amblyomma americanum and in Dermacentor variabilis in Missouri. And that there are also reports from Texas on successful isolation of this spirochete from Amblyomma maculatum, Amblyomma americanum, and Ixodes scapularis, as well as from pools of the cat flea Ctenocephalidis felis. Although Lyme disease has been reported from practically all the lower 48 states, endemic areas are in the northeast, upper Midwest, and in northern California. Indeed, it may well be that the Lyme disease spirochete or closely related spirochetes are occurring throughout our country, and in some instances in as yet unrecognized enzootic foci, where they are maintained and distributed by ticks or possibly other blood-sucking arthropods that do not attack humans. Therefore, such foci would not come to the attention of health authorities until introduction of anthropophilic arthropods, either through birds or through domestic animals, leads to the transmission and to disease outbreaks. In support of these speculations are, number one, the presence of Borrelia burgdorferi in rabbit ticks, Hemophysis lepus palustris, Ixodes neotoni, and Dermacentor permapertus. All of these species have been found infected with Borrelia burgdorferi or similar spirochetes. And also past or current spirochetal infections in rabbits in northern California. The second point is that the isolation of Borrelia burgdorferi from 71 of 168 Ixodes dentatus that have been taken of naturally infected cottontail rabbits in New York City also supports this hypothesis. And finally, the claim that in Northern California Borrelia burgdorferi is maintained in an ecological cycle between the dusty-footed woodrat, a Neotoma species, and its tick Ixodes neotoni. All of the western black-legged tick Ixodes pacificus has been recognized as the primary vector of Borrelia burgdorferi to humans. It is of limited significance in the ecology of the spirochete. Ixodes neotoni, the woodrat tick, is said to have a broad enough host range to allow for shifts to alternative hosts upon which Ixodes pacificus becomes infected and then transmits the spirochetes to humans. And this is for Dr. Masters. Indeed, a similar situation may exist in your area and in other southeastern states. The Lone Star tick Amblyomma americanum, like Ixodes pacificus in California, may be an unimportant factor in an enzootic cycle of the spirochete, a cycle that may involve as yet unidentified animal hosts such as birds, rabbits, and so on, and their tick vectors. Now let us turn to human babesiosis, Babesia microti, the agent. Sharing geographic, I should show you where this is fluorescent antibodies white with the Babesia in the erythrocytes. Sharing geographic distribution, animal hosts and tick vectors with those of the Lyme disease spirochete is Babesia microti that I've shown you, the causative protozoan agent of human babesiosis. Here is Peromyscus leucopus, the host of Babesia microti, and as you all recognize, Ixodes scapularis, the transmitting agent. The first known outbreak of this tick-borne disease occurred on Nantucket Island in Massachusetts in 1975, and it involved 6 persons. Prior to this outbreak, only 7 cases of well-documented babesiosis were known in the world, 2 from Nantucket Island, 2 from Yugoslavia, and 1 each from Ireland, California, and Georgia. The causative agent, a protozoan parasite of blood cells, was identified in all Nantucket patients. And it was soon established that the major reservoir of this parasite was the white-footed mouse Peromyscus leucopus, and that the deer tick Ixodes scapularis was the primary vector. Since the outbreak on Nantucket Island, additional foci have been recognized in other areas in Massachusetts, in Rhode Island, New York, and Connecticut, California, and in Wisconsin, the areas where you also find Lyme disease. Babesiosis is a malaria-like illness characterized by fever, shaking, chills, drenching sweats, myalgia, arthralgia, extreme fatigue, and a moderately severe anemia. Because asymptomatic infections are common, many cases are detected only through several surveys. Thus, there are no data on the prevalence of this illness, of which the majority of known cases occurred in the state of New York. A recent paper describes the epidemiology of 136 cases reported from 1982 through 1991, for which a detailed case reports are available. Accordingly, 103 patients, 76%, had to be hospitalized, 7 died, 2 suffered also from Lyme disease. Based on the experience in New York, several parallels can be drawn with Lyme borreliosis. The geographic distribution of both diseases coincide, and both Babesia microti and Borrelia burgdorferi share the same reservoir animal and the same tick vector. It is therefore not surprising that dual infections have been recorded in white-footed mice as well as in ticks. The latter, the ticks, have been shown capable of transmitting both agents simultaneously, spirochetes as well as Babesia. Now let's turn to Rocky Mountain spotted fever. This is a CDC slide. The second most prevalent tick-borne disease in the United States is tick-borne typhus or Rocky Mountain spotted fever. It is still a measurable health problem, even though the number of yearly reported cases has dropped from the all-time high of 1,200 cases in 1981 to less than 500 cases in 1992. As yet, we have no explanation for this nationwide decrease. Undetermined population events of rickettsia and the ticks affecting the causative agent rickettsia have been cited. One may also speculate that educational material on Lyme disease and its tick vectors may have increased the awareness about ticks in the last few years and about the steps to be taken to prevent unnecessary exposure. It is sad to report that the fatality rates of spotted fever still runs between 3 and 8%. Pretty high for a disease that can be treated effectively with antibiotics. The deaths occur because patients do not seek proper medical care, the disease is not recognized, particularly in its early stages, or because specific treatment is delayed. The diagnosis of spotted fever on clinical grounds before the appearance of the rash without a history of tick bite is indeed difficult because the disorder resembles a variety of febrile illnesses without localized manifestations. Involvement of the central nervous system, common in children suffering of spotted fever, often leads to the suspicion of acute bacterial and viral meningoencephalitis. Also, many physicians are unfamiliar with the pathognomic petechial rash, which in most cases appears characteristically on the palms and soles and should indicate immediate treatment with broad-spectrum antibiotics. Unfortunately, in about 11% of cases, the disease may take its course with a fleeting or atypical rash or with no rash at all. In such cases, a diagnosis is very difficult, with clinicians having to rely primarily on their knowledge of the right spectrum of the illness to identify patients with possible or probable spotted fever. Empirical anti-rickettsia therapy is recommended before verification of the diagnosis. Without this therapy, patients may die. As properly stated by Dr. Sexton in one of his recent publications, and I quote, "Recognizing spotless or almost spotless spotted fever is analogous to seeing a wolf in sheep's clothing. Intuition, experience, knowledge, and a high index of suspicions are required." Rocky Mountain spotted fever is a bacterial disease caused by the obligate intercellular in the blood cells of infected ticks. It is east of the Mississippi. The Lone Star tick, Amblyomma americanum, this is the distribution variabilis. The Lone Star tick, Amblyomma americanum, has been incriminated as a vector, but to the best of my knowledge, there has never been a case of Rocky Mountain spotted fever resulting from the bite of this tick species, even though spotted fever group rickettsia, non-virulent for humans, have been recovered from Amblyomma americanum. The focal nature of spotted fever, particularly its sudden presence in areas where the disease has never before been recognized, is a long-known phenomenon. Thus, the recent outbreak in 1987 in the Bronx of New York City did not come as a surprise. It should be recalled that none of the 4 patients had traveled outside the city within the 3 weeks before onset of illness. One patient, the only one bitten by a tick, died, possibly because the diagnosis and the treatment were delayed. The presence of spotted fever group rickettsia infected D. variabilis in the Soundview Park, frequented by at least 3 of the other patients, and the absence of ticks in 8 additional parks throughout New York City suggested the establishment of a focus, possibly through a dog, introducing infected ticks from the neighboring Suffolk and Nassau counties, areas endemic for spotted fever. Indeed, it is well known that man's best friend, the dog, is the most important means by which such transfers are accomplished. Dogs are of no significance as the sources of rickettsia for infecting the ticks, since only the adult stage of this tick feeds on dogs. On the other hand, there is ample evidence that spotted fever may be contracted through manual deticking of dogs or as a result of contamination with rickettsia-laden tick feces during petting dogs. 2 of the New York patients with no history of tick bites did have close contact with dogs that regularly visited the Soundview Park. Dogs ill with spotted fever rarely develop clinical disease. The experimental infection of dogs, on the other hand, has resulted in disease similar to human spotted fever. It appears that the severity of this disease in dogs is rickettsia dose related and may range from febrile exanthema to death. However, there are recent reports now of clinically severe spotted fever in dogs with clinical symptoms including depression, anorexia, multifocal neurological signs, and petechia of the mucous membranes, suggesting the emergence in certain geographic areas, especially in the south and southeast, of rickettsia strains that are unusually highly virulent for dogs. There is no vaccine against spotted fever, although the studies have shown, molecular and immunochemical studies are underway in preparing a vaccine that eventually could be used for humans. An experimental vaccine has already been developed and is very effective in laboratory animals, but it will take years before it can be applied to human use. A disease of dogs difficult to differentiate clinically from spotted fever, especially during early stages, is canine ehrlichiosis, caused by the leukocytic rickettsia Ehrlichia canis. You can see right here in this leukocyte the Ehrlichia. This is an electron micrograph showing you what's called a morally, an assembly of Ehrlichia canis agents. Canine ehrlichiosis is a febrile illness characterized by serious nasal and ocular discharges, anorexia, depression, loss of weight, and pancytopenia. In many instances, the disease is mild and may not be recognized. However, in some dogs, especially in German Shepherds, a severe, often fatal hemorrhagic episode occurs 60 or more days after initial infection and is associated with aplastic anemia. The causative agent Ehrlichia canis is transmitted by the brown dog tick Rhipicephalus sanguineus, in which it develops transstatally. In the United States, this tick has been reported now from almost all the states, and serologic surveys of dogs has identified the disease throughout the tick's distributional area. Rhipicephalus sanguineus in the United States rarely feeds on humans, which is very peculiar because down in Mexico, in South America, Rhipicephalus sanguineus feeds quite readily on humans. In 1976, a symposium on canine ehrlichiosis was held at the Walter Reed Army Institute of Research in Washington, D.C. After the session on tick transmission of Ehrlichia, I was asked to express my opinion concerning future problems and investigations related to this subject. I predicted epizootics throughout the distributional areas of Rhipicephalus sanguineus. They did not occur. And I referred to the possibility that other species of dog ticks, such as Dermacental variabilis and Amblyomma americanum, may become infected and involved as vectors of Ehrlichia canis, affecting not only dogs, but possibly also humans, and that did occur. The first case of human ehrlichiosis, initially thought to be caused by Ehrlichia canis, was diagnosed in 1986 in a 51-year-old man who had been bitten by a tick 12 days before onset while planting trees in Arkansas. His diagnosis was confirmed by the demonstration of Ehrlichia in affected leukocytes. Since this initial report, 296 additional cases of human ehrlichiosis have come to the attention of CDC. Most of these came from South Central states, with a few cases also from the West Coast. In the meantime, it was also established that the causative agent, although closely related to Ehrlichia canis, is a hitherto undescribed agent for which the name Ehrlichia chaffeensis was proposed. Here you see leukocytes again with the, let me see where I am here, with the agent throughout the cell. The symptoms for human ehrlichiosis are non-specific, and there's an electron micrograph, again very similar to the other Ehrlichia slide we have seen. They're very similar to those of mild spotted fever, including fever, arthralgia, myalgia, headache, anorexia, nausea, vomiting, chills, pneumonia, leukopenia, and in about 20% of the patients, a rash. Approximately 85 to 90% of patients had a history of tick exposure, 1 to 4 weeks prior to onset. Yet in none of these cases could the species of tick be determined. Until this is accomplished, we can only speculate as to the transmitting agency. The following thoughts should be considered. It may well be that the Epicephalus sanguineus, the brown dog tick, is beginning to attack humans. It is also possible that other species of ticks, such as Amblyomma americanum or Ixodes scapularis, are parts of the natural cycle of Ehrlichia. The American dog tick D. variabilis cannot be considered a potential vector because, as pointed out before, only adult ticks feed on dogs, and for them the dog is a dead end. Another recently emerged disease with a suspected possibly tick vector is equine monocytic ehrlichiosis, also referred to as Potomac horse fever, ehrlichial colitis, and acute equine diarrhea syndrome. This disease was first recognized in South Central Maryland in 1979, and today is known to affect horses throughout this country. So if you own a racehorse, there is a very economic problem. The disease is characterized by fever, depression, anorexia, leukopenia, colic, and mild to severe diarrhea. It is fatal to about 30% of affected animals. The seasonal nature of this disease, which is May through October, I think we are out of focus here. Let me get that back. Okay. This seasonal incident suggested that ticks may be involved in the transmission of the causative agent referred to as Ehrlichia rhisticiae, especially Amblyomma americanum or again Ixodes scapularis. It's also possible that biting flies and other insects are involved in maintaining and transmitting this agent. We just don't know as yet. Fortunately enough, the Potomac horse fever agent, Ehrlichia rhisticiae, is susceptible to tetracyclines, which have been shown effective in treating horses, especially when administered immediately after the onset of fever, and there is a vaccine, two vaccines actually on the market that are protecting horses from getting seriously ill. Tularemia, it continues to be a health problem, particularly in the southwestern part of this country. Of 1,733 cases reported nationwide during the 7-year period of 1981 through 1987, 63% or 1,041 cases were contracted in the southwest central region. Of these, 63% were tick associated, whereas 32% contracted the disease throughout or through handling infected animals, particularly rabbits. According to the Center for Disease Control, 150 to 250 cases of tularemia occur each year. Ladies and gentlemen, the recent emergence of new animal and human diseases in the United States provided and still provides exciting and rewarding research opportunities. Indeed, there is no doubt that many of the questions touched on today will soon be answered as a result of ongoing or scheduled meticulous laboratory and field research. Tick-borne diseases in the United States will continue to affect large segments of human and animal populations as long as we share our biotopes with those of ticks and their host animals, and as long as we skillfully, and I emphasize skillfully, create the ecological conditions leading to massive tick infestations. I want to show you just a couple more slides. This is the prevalence of tick-borne relapsing fever in the United States, and we have anywhere between 40 to 50 cases per year. Again, out of focus here. There. The distribution, the geographic distribution of this disease, you can hear throughout the Northwest. And the three vectors, Hermsite, Turricardia, and Parkeri. We talk about Colorado tick fever, which is limited to the Rocky Mountain areas. Every year, about 25 to 30 cases or 50 cases are occurring. There's nothing you can do against Colorado tick fever except take a few aspirins. It's a viral disease, stays in the erythrocytes for a long time. Patients suffering of Colorado tick fever are down for months, sometimes for a year, week. And this is the vector, Thermocenter andersoni. Then we have Colorado tick fever here, the reservoir, which is the golden mantle ground squirrel, Spermophilus lateralis. New disease entities will emerge and are already knocking at our borders. Thus, I would not be surprised if Calvea ruminantium, the causative agent of heart water, already present in the Caribbean, would establish itself on the mainland, where it could readily be maintained and distributed by the Gulf Coast tick Amblyomma maculatum. A tick experimentally shown to be an efficient vector of this serious disease of ruminants. Not an encouraging but certainly necessary thought for our continued struggle with tick-borne diseases. Ladies and gentlemen, I thank you for your attention. If there are some.
Can we have the lights, please? Yes. Briefly state how babesiosis is diagnosed and treated.
To the best of my knowledge, chloroquine is being used in treating babesiosis because it's a malaria-like disease. Diagnosis, demonstration of Babesia microti in the erythrocytes. We wish we would have something simple like that for Lyme disease, too. Any more questions? Yes, please. Have you anything, the question is, have you anything on concurrent infections of Lyme and babesiosis? Yes, there is in the literature are cases reported where patients are suffering of both at the same time. Because as I mentioned, the ticks can be infected with both, and they do transmit simultaneously both agents. Okay. We had some tangle illumination. Please, next questions? It's all clear. Thank you very kindly.
Thank you, Dr. The next session is going to be the new announcements, and what we'd like to do is, after each session, leave about 3 to 5 minutes for some questions, and hopefully we'll get some microphones in the audience. If not, just scream out and we'll try to hear the question. The next speaker is Dr. James Oliver. He's going to be discussing co-specificity of Ixodes scapularis and Ixodes damni, unmaining the deer tick. Dr. Oliver? Is he here?
No, I think it's okay. The projector's on, so all I have to do is hit forward.
Right.
Here we go. Yeah. Okay. Good morning. I give you now Professor Twist, a conscientious scientist. The trustees exclaim he never bundles and send him off to distant jungles. One day while camped by a tropic riverside, he missed his loving bride. She had, the guides informed him later, been eaten by an alligator. Professor Twist could not but smile. He said, "You mean a crocodile." This is a poem by Ogden Nash called The Purist. And I'm afraid a lot of people think that some scientists that worry about the naming of organisms may be a little over the edge. They may worry a little bit about inconsequential ivory tower type hairsplitting on what is A and what is B and what is C and so forth. And let me assure you that our investigations of the relationship between Ixodes scapularis and Ixodes damni has not been such an ivory tower approach, but has had practical reasons and significance, which I hope you will see as we go on into the lecture today. As you can see, the title deals with the relationships of Ixodes scapularis and Ixodes damni. We have a recently published paper where we synonymized those two species, published in the Journal of Medical Entomology as indicated on the slide. You also have that paper, I think, copied in its entirety in your booklet, as well as an abstract of that. In addition to our paper, which I'll go into in a bit more detail, there's a paper in press now by Don Wesson and other authors, and it essentially confirms our paper where we say that the scapularis and damni are the same species. Don in the group used the internal transcribed spaces ITS1 and ITS2 of the ribosomal DNA from Scapularis damni and pacificus, and they sequenced them and analyzed at 3 different levels within individual population variation, between individuals of different geographic origin within a species, and between species. And the conclusion is, based on maximum parsimony and 2 distant methods of analysis, sequence variation of ITS1 and 2 suggests that Scapularis and damni are not distinct species, and even individuals from geographically isolated locations are very similar. Individuals from geographically separated populations of Ixodes pacificus appear relatively less closely related to each other, but distinct from those of our Scapularis damni. So it's a confirmatory follow-up to the work that we were doing, and is based on genetic sequences. Now, what is the significance of our finding? Because most of you probably really don't care what something is called. Well, there are several different types of data that I think are useful. One is that significance may be that it might alert medical doctors and public health officials that if Scapularis is present, at least one prerequisite for Lyme disease transmission is present. Some data on Lyme disease and damni can be extrapolated from the north to the south, but ecology of different geographic populations of Scapularis will vary, reflecting local density dependent, meaning tick populations, meaning host species, meaning numbers of host species, those types of density-dependent factors, and independent factors such as climate, photoperiod, and local factors of that nature. And geographic strains of Borrelia burgdorferi are thought to be probably a good potential to vary in different regions as well. Our work was based on an approach to seeing the relationships between Scapularis damonii and pacificus. Now, most people describe a species based on its morphology or its form. And this is what museum taxonomists have to do. This is the specimens that they have to work with. People send in specimens, they're in alcohol, they look at them, and they see if this one looks different from this one, and it seems pretty consistent, then we call this one species A, and we call this one species B. That's fine. That helps. That's what they have to do. But on important problems of genetic relationships or species separateness, one would want to do more to really find out. Because look at the variation, for instance, in this room. We're all the same species. Go to the American Kennel Club for a show and look at the differences in morphological forms in the same species. So morphology is a good first guess as to what a species is, but it should not be taken as the ultimate test unless you don't have any other material, which again, in probably 90-something% of the cases of species, museum taxonomists don't have any else, so they do with what they can, and they provide a very valuable source. But on species such as this, it has very important medical and veterinary importance. One would like to go further. Also, because the Ixodes damonii deer tick terminology is so entrenched in the literature, and it is such an important problem, we felt we had to go to greater extent to try to prove whether they were or not the same species. One of the reasons that we were told that Lyme disease did not occur in the South, or if it did only rarely, was that the species were different. So this is one of the reasons we go about it. You can see, number one, morphometrics. That's the thing that I've been talking about in morphological form, but we've gone in greater depth. We've been able to analyze many more characters than most approaches, most taxonomists do. We've also used isozymes. We used DNA sequencing, which you saw was a continuation that Don Wesson and authors did. We looked at the chromosomes of these different populations. We attempted to hybridize each of these with each other. That being one definition of a species is reproductive isolation. Then we said, well, okay, if they hybridize, will we get criticized for saying yes, but the pigs were in a sexually lovable mood and they didn't give them any choice, so what were they to do? Well, we give them choices. Finally, host specificity has often been cited as a difference between the northern and southern Ixodes scapularis population. And I don't believe that's true. In my talk tomorrow, when I talk about borreliosis in the southeastern United States, I can show you data that show when you bring these now two species into the lab and offer them hosts, you can't distinguish even between Pacificus and the eastern species on the basis of what they would choose to feed on. And there's many other questions about the tendency for the northern population to feed on humans, where the southern population doesn't feed on humans. We'll get into some of that tomorrow as well, because I want to see the data that show that. Since 60 or 70% of the patients in the Northeast never remember being bitten by a tick anyway, there's a lot of questions involved here. So I'll talk more about host specificity in tomorrow's talk. In our studies, the colony origins were, as you can see, Ixodes damonii were from Great Island in Massachusetts, and Andy Spielman was kind enough to send us populations of that. We collected Ixodes scapularis in Statesboro, Georgia area, and Pacificus was sent to us by Bob Lane at the University of California, Berkeley, and he collected it at Point Reyes National Seashore over in Marin County, just north of the Golden Gate Bridge. So that basis is the genetic basis of spot that we started working with. In the morphometric study that I mentioned, instead of using 5, 10, even 20 characters that most taxonomists have to work with, one of my graduate students, Joel Hutchison, who is working on his doctoral dissertation now, is able to use a multivariate statistical analysis approach where he is able to use many more characters, and presumably using more characters would give us a better quantitative and real difference in the morphological variation within the species. And these are the areas from which he has colonies in the lab. I think there's 17 different geographic colonies he now has growing, and from that took measurements of the F1 from these so that the measured specimens would not be influenced by the environment, i.e. you get differences depending on the state of engorgement and that type of thing. So we try to eliminate that variable. He would use various analyses, but one I'm going to show you today because of the scarcity of time is just one, where he used size-free discriminant analysis and it involved the following procedures. Well, I don't want to go through all of this because again time is too short, but I'm going to show you one scatter plot generated from using, again, size-free discriminant analysis. This is from nymphs of 4 naturally occurring populations from Massachusetts, from Maryland, from North Carolina, and from Georgia, and 2 populations of hybrids, reciprocal hybrids from Georgia and Massachusetts. The Pacific coast tick, Ixodes pacificus out here, unfortunately it's purple, doesn't show up very good in this room, but let me outline it for you right there. That's Ixodes pacificus. These other dots represent individual nymphal ticks. And we use nymphs here because of all the 3 stages of ticks, nymphs are the ones said to be the easiest to discriminate between, say, Massachusetts and Georgia ticks. And in fact, that's true. And in fact, we can still discriminate some differences if we take certain characters, morphological characters, and look at it from our Massachusetts colony and our Georgia colony. But what happens is there's a gradation as you go up and down the East Coast, and we're looking for East-West as well in decline. You can see gradations of these characters where if you take a tick out of maybe North Carolina, Virginia, or somewhere in between, those characters are not so distinct. It kind of merges in together. And you can see how these are overlapped with each other. That's Massachusetts there. Georgia is here. Georgia, Massachusetts. And North Carolina is the green. The Maryland is the white, and there are the hybrids in between. So there's much overlap in morphological characters. So people picking up ticks in the field have a very difficult time saying, well, is this a Damini or a Scapularis? Well, the answer is that they're the same species because they interbreed, and I'll show you the other data in a minute. But even the morphological characters grade as you go in these 2 different areas. Let me emphasize again, we can still recognize using a nymphal stage, using particular characters, recognize some ticks from Massachusetts and some from Georgia. But that in no way means that they're the same species. And you can't always do that, as a matter of fact. In our hybridization attempts, as you can see here, we put males and females of similar ages placed together in vials 24 hours prior to putting them on a rabbit, and then we fed them. And then as we got offspring, we monitored them for oviposition, hatching, molting, tick weight, all this kind of thing. When we did the crosses, this is between Ixodes scapularis from Georgia and Ixodes pacificus from California. You can see that hybridization between them, both the reciprocal cross, when scapularis was the female and when pacificus was the female, they did produce progeny but the F1's progeny were all sterile, and you can see that fertility, and this is egg mass or fraternity here. So Scapularis pacificus will produce F1, but the F1 are all sterile, so that's the end of the line. When you cross Damini, formerly Damini from Massachusetts, with pacificus, again, you get progeny, you get F1 produced, but they're sterile, so that's the end of the line there, constituting a good reproductively separate species. continue across, you find a Gamma from Massachusetts and Scapularis from Georgia. You not only get F1s that are fertile, 94%, and you do the reciprocal, 72%, but you take it to the F2, and they're fertile as well, and we've taken it to the F3 generation now, and they're still fertile. So we quit the experiment at that point. So you can see that the Massachusetts and Georgia populations clearly produce offspring when they come together. So the criticism could be leveled at, well, again, if you don't give them any other choice, and they're in a mating mood, they're going to mate with whatever you put it in. It's a forced laboratory experiment that you're running. Well, that's not so. We did this by what's called a selective mating, or choice mating. And in this situation, what we did was first determining successful mating was determined by opening up the females and seeing if we could find sperm in the females. That was a positive fertilization. Each group consisted of 4 replicas of 10 each, and there was a limited sex in each one. For instance, with the Gamini, we did female choice and the Scapularis female choice, and the Gamini male choice and Scapularis male choice. In other words, we put 10 female Gamini in with 10 male Gamini and 10 male Scapularis, so that each of these females or males had a choice, an equal number of partners of the same population and of 10 out of the other population to see if they would choose to mate with their own if they had the opportunity, while others were present. And you can see here that female Massachusetts ticks, 16 out of the 40 mated with Massachusetts males, 19 mated with Georgia males, and 5 didn't mate at all. Of the Gamini males, when they started having a choice, they indeed did seem to recognize their females, and 14 out of 40 mated with the same population, 4 with the Georgia males, and 22 for some reason didn't mate at all. When you get the southern population of choice, female of choice, she also showed preference to the males of her group over the Massachusetts population. 5 didn't mate. And Scapularis males actually preferred Massachusetts females over the Georgia females. So you see, it goes all kinds of ways. And clearly, even in the ones that preferred or recognized their own kind, that was mating of the other kind as well. So if these ticks were to come together in nature, and there'd be mixtures of the different populations, clearly they would behaviorally mate. And we've already shown that genetically that would be a successful experience. The chromosome analysis was a little unrewarding in that at the levels we investigated, we could find very little differences. All 3 of these populations had a diploid number of 28. The sex chromosome mechanism was the same in all of them. Relative lengths of chromosomes at mitotic metaphase, same in all of them. We did find in seed-banding analysis that Massachusetts and Georgia ticks possessed an additional interstitial seed band on chromosome 7, and this band was absent in the Pacific Coast species. We wanted to look at isozyme analysis, since many systemicists or taxonomists are using that now as an indicator of genetic relatedness. And you can see what we've done here. We've used Nye's Genetic Identity Index. First, you should notice that values above 0.85 are considered to reflect conspecificity. And you'll note when we compare Massachusetts and Georgia, we get a 0.9, which is above the 0.85. So it does indicate conspecificity. When you compare Pacificus and Scapularis, you get a 0.3, Gamini and Pacificus a 0.4, and this is a control. We compared Massachusetts and Georgia ticks against Denisona variabilis, and they had a very low genetic identity outside of the conspecificity level. According to Nye and Ayala, the ranges in the figures that we got between Pacificus and Scapularis and Gamini and Pacificus were in the range of congeneric. That's in the range of conspecific, range of congeneric, and this is outside of that. So again, another approach, another way of getting at what the relationship of these ticks are. This is part of a bigger investigation that we're conducting on genetic relationships of the ticks in the Ixodes ricinus complex, or as the Russians would say, Ixodes persicatus complex. We wanted to understand more about all these ticks, but in particular, the ones that are proven Lyme disease vectors, such as Pacificus and now just Scapularis. As you know, we synonymized that. And in the Palearctic area, where you have Persicatus and Ricinus as the major vectors. We now have a colony of Aphanus. We're just colonizing now, and we hope we'll be able to check that as far as vectorability of the Borrelia spirochetes is concerned, and also genetic affinity to the others. Our goal is to compare those four major vectors in the world, again, to see how closely they're related, to get ideas of the potential evolution of these things and where maybe these ticks originated from, as to whether the ticks in the North American continent came from Europe or Asia and its relationship. But that's too involved right now. And we're doing this. We're using DNA comparisons, and we're requiring DNA of these species from various world populations. And again, Persicatus, Ricinus, and the various different geographic populations of Pacificus and Scapularis to permit molecular genetic assessment of systematic affinities. Life cycle, people say there's a big difference in life cycle between Damini and Scapularis, at least they used to say that. We found no differences when these ticks were brought into the laboratory, given the same photoperiod, given the same host, given the same temperature, and so forth. We haven't analyzed all of the parameters yet in the life cycle. We're in the process of doing that now. But what we see is that when you bring them in the same conditions, they react similarly. Now, of course, the population in the north has a 1.5-year to 2-year cycle, and we think that the population in the south has a 1-year cycle. But again, this is environmentally triggered. If you bring them into the same environment again, you get the same type of cycle. Vector competence. There's been at least a couple of papers, Willie Bergdorfer and Gage and Joe Peisman and Sinsky. They've both shown that Scapularis, or I should say 7 populations, are fully competent vectors. And in addition, Dr. Bergdorfer tells me personally that in the laboratory, he can't distinguish any difference between vectorability of the northern and southern population. So again, you can't separate these things out on vectorability. So in conclusions, Scapularis and Damini are conspecific. The name Scapularis has priority over the name Damini according to the International Code of Zoological Nomenclature, and therefore the species should be called Scapularis and not Damini. Common name of Scapularis is the black-legged tick, not the deer tick. Now, I don't know how this is going to go with all the textbooks and all the people, and as Willie said this morning, it's such a habit to say Damini. It's so easy, and it's so easy to say the deer tick. And really, I could get into a little 2 or 3 minute talk about why we should call this, why we shouldn't call this thing the deer tick, but I won't get into that because it's so convenient for people to do. But it does suggest that a role of the deer greater than it does, although it's tremendous in producing the number of ticks and increasing the population, it is not thought to be a competent reservoir for the spirochete. So at any rate, I don't know how long it'll take place, and I really don't personally care if you people want to continue to call it whatever. I just want people to know that the tick is the same, has the same vector potential, and that's the point I think is important for this group, is that now it opens up the possibility of regions where Scapularis is present. It has the potential, at least one of the factors in the equation of possible human involvement, that it is there. And I would argue with people about the difference in the habits of the propensity for the southern population to feed on humans versus no. I'd like to see some more data on that. Thank you very much.